We are going back today, way back to the very beginning of, well, what became the defining public health crisis of our time. I mean, think back to those first few months of 2020. The world was just starting to grapple with this new virus, SARS-CoV-2, and how fast it was spreading. You had headlines, what, by early March? Over 80,000 cases, 3,000 deaths in China alone, and it was clear this was going global. And the biggest question wasn't just where, but how. How does this thing actually get inside a human cell? So for this deep dive, we're going straight to the source, a foundational paper from that time that mapped the infection's very first step at the atomic level. We want to understand that lock and key mechanism.
Right, and that lock and key idea is really central to this whole family of viruses. We're talking about the beta coronavirus genus. So that includes the original SARS from 2003, MERS, and of course SARS-CoV-2. They all use this massive structure on their surface. It's a homotrimeric spike glycoprotein.
It's like a three-part antenna.
Exactly. And that spike's entire job is to find one specific thing on your cells, the ACE2 receptor, angiotensin-converting enzyme 2. That's the front door.
Okay, let's unpack that. Because if binding to ACE2 is that first critical handshake, then getting a picture of that handshake, I mean, an actual 3D map, that's everything. And these researchers, they did exactly that. They mapped where every single atom touches. Why is that level of detail so crucial? It's not just, for curiosity's sake, right?
Not at all. That map is the bullseye. It's the blueprint you need to design anything to stop it, whether that's a drug to block the lock or an antibody to cover the key. So to get there, they focused on the part of the spike protein that does the actual work. It's called the receptor-binding domain, the RBD.
The RBD.
Yep, it sits on the S1 subunit. And first, they had to prove it. They took HeLa cells, standard lab cells, and the ones that had human ACE2 on their surface could be infected. The ones without it, the virus couldn't get in.
So that confirmed it. ACE2 is the only game in town.
It's the necessary target.
And the method they used to get this map, I mean, this is just classic structural biology. The source says they used X-ray crystallography.
You basically create a crystal of the two proteins locked together, the viral RBD and the human ACE2, and you shoot X-rays at it. The way the X-rays bounce off gives you this incredibly detailed 3D map.
And the detail here was what, 2.45 angstrom?
Yeah, 2.45 N.
Which, for anyone listening, an angstrom is 1/10,000,000th of a meter. So we are literally seeing the positions of individual atoms. That is a fantastic resolution.
It is. And once they had that map for SARS-CoV-2, they could put it right next to the one that already existed for the 2003 SARS-CoV. And this is where it got really fascinating.
What did they find?
They looked almost identical, even though the genes that code for their spike proteins are, less than 80% the same.
Wow.
The whole binding strategy, the way the new virus's RBD grabs onto ACE2, is nearly a mirror image of the old one. The researchers saw this as, well, a classic case of convergent evolution.
Meaning that nature sort of found the one best way to solve a problem, and both viruses stumbled upon that same solution independently.
Precisely. It's like two different engineering teams working decades apart, both designing a key for the same very complicated lock, and they come up with the exact same shape.
And the numbers back that up, right? The root mean square deviation, which is a measure of how similar they are, was only 1.2a.
Which is incredibly close in molecular terms.
And even the contact patch, the buried surface area, was basically the same. SARS-CoV-2 covers 1,687 square angstroms. SARS-CoV was 1,699. It's the same amount of real estate.
And that contact happens at a specific part of the RBD, a little loop called the receptor-binding motif, or RBM.
So if the RBD is the hand, the RBM is kind of like the fingertips that actually make contact.
That's a great way to put it.
Okay, so this is where it gets really, really interesting for me, because we all know how this story played out. The 2003 virus was contained. The 2020 virus caused a global pandemic. If the keys look the same, the contact area is the same size, the difference has to be in the tiny details, in the individual amino acids.
Exactly. You have to zoom in from the blueprint to the individual rivets. So both viruses contact about 20 residues on ACE2. And of the 14 key contact points in the RBM, 8 of them are identical. So there's a huge amount of conservation there.
8 are identical. What about the others?
5 others have similar types of residues, maybe one bulky, one swap for another, but the actual side chains are a different shape, and those tiny tweaks start to change the fit.
So we have a lot of similarities, some minor tweaks, but what's the smoking gun? Is there one single change that really made a difference for SARS-CoV-2?
There is, and it's actually a residue that's just outside that main RBM contact area. It's a residue called lysine 417, K417.
And lysine 417.
In the SARS-CoV-2 structure, this lysine is perfectly positioned to form a salt bridge with a residue on the human ACE2 protein.
Okay, a salt bridge. Let's pause on that. That's a really strong electrostatic bond, right? Between a positive charge and a negative charge.
Exactly. Lysine is positively charged, and the residue it interacts with on ACE2, aspartate 30, is negatively charged. They
Huh.
snap together like two powerful little magnets,
Creating an extra anchor point.
a very strong one. It
Mm.
creates this unique positively charged patch on the surface of the new virus's RBD that the old one just didn't have.
And what was in that spot on the 2003 virus?
It was a valine, V404. And valine is neutral. It's hydrophobic. It didn't interact with ACE2 at all. It just sat there.
Ah, so that's the upgrade. You swap a neutral, non-participating residue for a positively charged one that forms a strong salt bridge. You've added a powerful magnet to the key. So what did that do to the virus's sticky
It's
edge?
translated directly into a huge jump in binding affinity. We measure that with something called the dissociation constant, or KD. And with KD, the smaller the number, the tighter the bond.
And the numbers were
For SARS-CoV-2, the KD was a really potent 4.7 nanomolar.
4.7.
For the original SARS-CoV, it was 31 nanomolar.
31 versus 4.7, that's more than a six-fold increase in binding strength. And that comes from just a few little atomic changes, but especially that lysine salt bridge.
It's a major reason for the increased efficiency. But, and this is a really important point the paper makes, that tighter binding alone probably isn't enough to explain the incredible transmissibility we saw.
Wait, really? That seems counterintuitive. A stronger lock and key connection seems like it should be the whole story. Why not?
Well, the paper suggests that transmissibility isn't just about getting into the first cell efficiently. It's about replicating and spreading from person to person. And for that, we need to look at another unique feature of SARS-CoV-2,
Okay.
the RR furin cleavage site.
The what?
It's a sequence of 4 amino acids, arginine, argine, alanine, arginine, right at the hinge point of the spike protein between the S1 and S2 subunits. And it's a perfect target for an enzyme we have all over our respiratory tract called furin.
So our own body's enzymes are basically priming the virus for infection.
That's the hypothesis. This cleavage is thought to make the virus much, much better at fusing with our cell membranes. So the tight binding from the K417 change gets it in the door, but this furin site helps it replicate and spread like wildfire.
Wow, what a combination. A super sticky key and a self-priming engine. Okay, let's shift gears to immunity. With all this structural similarity, you have to think there was enormous hope that antibodies from the 2003 SARS survivors would work against this new virus. Did that hope pan out?
Unfortunately, no, not really. The initial results were, well, pretty disappointing.
How stab.
While the raw serum from recovered patients, so their whole mix of antibodies did show some cross-neutralization, the specific high-powered monoclonal antibodies that have been developed for SARS-CoV mostly failed.
They just didn't work.
Many of them showed no notable binding at all. And it's a huge lesson in how precise this all is. The overall key shape was the same, but the specific molecular address the antibody was designed to find had changed just enough.
And we can link that back to those residue changes we talked about.
Absolutely. The paper gives a great example. There was an antibody for SARS-CoV called ADR. They mapped its binding site, its epitope. On SARS-CoV-2, that exact spot had 16 residue changes out of a total of 25 positions.
16 out of 25.
Yes.
That's incredible. So almost 2/3 of the contact patch that antibody was trained to recognize was completely different. It's like trying to use an old key in a new lock where they've changed most of the pins.
No wonder it didn't bind. It shows how quickly that part of the virus, the RBD, can evolve to evade our immune system. There was one bright spot, though. One antibody called CR3W22. It was developed for SARS-CoV, but it did bind to the SARS-CoV-2 RBD, and it bound tightly, a KD of 6.2 nanomolar.
So that proved that cross-reactive sites do exist. They're just maybe harder to find.
Exactly. It gave researchers a path forward.
So for you listening, this is where the practical application comes in. This structural map wasn't just an academic exercise. It became the blueprint for defense.
Absolutely. The takeaway for scientists at the time was clear. Future vaccines and antibodies should probably focus on more conserved epitopes. The RBD, that sticky part, is just too variable.
So instead of aiming for the part that's always changing, you aim for the parts of the engine, maybe on the S2 subunit that the virus can't afford to change. The parts that are shared across lots of different coronaviruses. Okay, so to sum this all up for you, at the atomic level, we saw that SARS-CoV-2 used a very similar playbook to its 2003 predecessor. That's the convergent evolution. But it came with a critical upgrade, a much stronger grip on our cells, thanks in large part to that unique lysine 417.
And that detailed structural knowledge, published right at the start of the crisis, became the absolute bedrock for designing the vaccines and the antibody therapies that followed. But here's the thought I want to leave you with. If these two different viruses, separated by almost 2 decades, both independently evolved such a similar way to attack the human ACE2 receptor, what does that tell us? It suggests there are really tight structural rules for how this family of viruses has to adapt to jump to humans.
That if another one comes along, it might use a very similar-looking key.
It might. And the question is, what parts of that key absolutely cannot change? If we can figure that out, we might just be able to build a master lockpick for the next pandemic, something to really mull over.